it is a great pleasure to introduce Dr. Mark Hildebrand. Now, Mark started his scientific career at the State University of New York in Syracuse, where he received his BS in chemistry. And then he changed fields slightly. He moved west to the University of Arizona, where he did his PhD in biochemistry. And he finished that in 1987. And then he came to Scripps to do his postdoctoral research. And he was a scholar here working with Dr. Benjamin Volcani. And he stayed. He must have liked it here a lot. He's currently an associate project scientist in the Marine Biological Research Division, where he conducts some extremely interesting research. It's very highly, in fact, cross-disciplinary, and it involves research work in the fields of diatom molecular biology, genomics, biotechnology, nanotechnology, among other areas. He's really a very varied researcher and does an awful lot of different things. When I was a child growing up, I always called diatoms diatoms. And I'm having great difficulty not saying that tonight, so I thought I'd tell you. Diatoms are really incredible single-celled organisms, and they have cell walls made of glass. And they've always intrigued me because the patterns are so beautiful and so intricate. And as Mark will tell us this evening, they're also incredibly useful for all sorts of applications. So without further ado, let me introduce Mark and say that he's going to talk tonight on the beauty of diatoms from the marine food web. Okay, the beauty of diatoms from the marine food web to nanotechnology. Thank you.
Thank you, Nigella. Thank you all for attending. Now I'm going to have trouble pronouncing that word. No matter how you pronounce it, diatoms are really incredible organisms. And what I'm going to talk about tonight are some of the basic cell biology of diatoms, which probably many of you are familiar with. But just to review why they're interesting organisms. And then I'll expand the topic a bit to talk about why we're interested in diatoms in a global sense, in the sense that they're very important organisms as a basis of the marine food web. And then talk a bit about a very exciting area, a new area of diatom research. There's a lot of interest in this right now, and it has to do with the possible application of diatoms as the source of materials for nanotechnology. So let's start now by introducing diatoms. They're unicellular photosynthetic algae. And this is a light micrograph of two individual diatom cells. You notice the green pigment, which is chlorophyll. One of the distinctive features about diatoms is that they come in an incredible variety of shapes. And this micrograph here is showing individual and distinct diatom species. And in fact, the estimated number of diatom species globally is in the tens of thousands. And each species is characterized by a unique shape. The diatom cell wall is also somewhat unique in the sense that it has an organic component to it. So if you look at an untreated diatom cell, you can see the sort of amorphous structure to it. If you clean the organic component away with acid treatment, however, you see the very distinctive feature of diatoms in that they form a major portion of their cell walls out of silica. And these organisms have evolved the means to take up silicon from the environment in a soluble form as silicic acid. It's dissolved in the water. They can transport it into the cell using specific transport proteins, which I'll talk about later. And then during the period of cell wall synthesis, they can catalyze the polymerization of silica to form a major portion of their cell walls. And as Nigella has mentioned, silica is the chemical composition of glass, and so diatoms are organisms that literally live in a house of glass. And this process of taking a soluble form of silicon from the environment and then mineralizing it through the intermediary of a biological system is called biomineralization. In the case of diatoms, it's biosilicification. And diatoms are the predominant organisms on the planet who carry out this reaction. How big are diatoms? Well, a typical diatom is on the order of 10 to 20 microns across. And this can vary quite a bit. There are much larger diatoms up to millimeter size, but most are in this size range. And so in relationship to a dime, that's a diatom right there. It's about 1/1000th of the diameter of a dime. And yet, even though this is small, if you look at some of the smaller scale features that are found in a diatom cell wall, they're much smaller. So they're on the order of tens to hundreds of nanometers. There are two general classes of diatoms, and they're classified both according to their structures and also evolutionarily. The first diatoms to arise in an evolutionary sense are called centric diatoms, and they're radially symmetrical. So if you take a point in the center of the cell, there's radial symmetry around that point. And it's not necessarily perfect symmetry. In this example, you don't have a mirror image on both sides of the center, but there's a general symmetry about the central point. And most centric diatoms are circular. However, there are variants of that. There are other centric diatoms which, again, are symmetrical about the center but not circular. The second class of diatoms are the pinnate diatoms, and these are laterally symmetrical. So if you draw a line along the length of the cell this way, they're symmetrical across that axis. And even within this constraint, there's a tremendous amount of variation in the actual shape and structure of these particular diatoms. But again, they're all symmetrical about the central axis. This one in particular is very interesting because it actually has helical symmetry. There's actually a helical structure in the cell that wraps around it. Now, the overall structure of a diatom can be visualized most appropriately as a petri dish. And this is an actual electron micrograph of a diatom cell in which the two halves of the cell are slightly ajar. And literally, there's an upper and lower portion to the cell that overlap each other. And this is drawn in cross-section right here. The upper portion of the cell is called the epitheca, and then the lower portion is the hypotheca. The structures on the top and the bottom, and these are what you've been seeing in the micrographs before, are called the valves. And then the valves are connected to each other through a series of overlapping girdle bands, which oftentimes span the circumference of the cell. This is a cutaway view of a pinnate diatom species, and we can see the valve on the bottom and the top. The pores in the diatom valves are called areolae, or pores. And pinnate diatoms generally have another structure in the center of the cell called the raphe. And oftentimes this is broken into two parts, although there are variations on this. And this is very important for these diatoms in the sense that it provides a mechanism by which they can actually move on surfaces, and I'll show you that in a couple of slides. When diatom cells divide, they do so as follows. This is a single mother cell diatom with a nucleus inside. The DNA is replicated and cytokinesis occurs, and you form two daughter cell protoplasts still within the mother cell. And then the new cell wall, or the new valve, is synthesized on adjacent faces of the two daughter cells. And the valve is made completely within the cell. And then there's a couple of variations that can occur depending on the diatom species. In some species, the valves are exocytosed. So they're made completely within the plasma membrane of the cell, but then the entire valve is moved to the outside of the plasma membrane. This is really an extraordinary feat if you consider this. The entire diameter of the cell, this large piece of the cell, is being moved from being within the cell to outside the cell. We don't really understand the details of how this happens. Once that occurs, these particular species of diatoms can then separate. The two daughter cells can separate. In other species, the valves are exocytosed, yet the two daughter cells are still attached, and they synthesize a new set of girdle bands. And then these can then separate, eventually going back to reforming the initial mother cell. Now, this process of division in diatoms is called vegetative division. The cells don't have to go through a sexual cycle. In fact, all diatoms go through this process where they can vegetatively divide. There are some diatoms, however, that are constrained to go through the sexual cycle. And this is actually a very old observation by two independent researchers, McDonald and Pfitzer, back in the 1850s, who noticed that diatom cells that had very rigid theca actually reduced in size with each generation because of the constraints that were imposed by the rigidity. So for example, if this is our mother cell with a theca of size A and then size B, after the first generation, theca A will generate another copy of B, but theca B will generate a copy of a smaller size C, If you follow this through to a second generation, you'll end up with 1 theca of size A, 3 of B, 3 of C, and 1 of D. And as you can see, the cells on average are reducing in size. Obviously, there's a finite limit to this. They can't just shrink away to nothing. So what happens is, when they reach a certain small size, it then triggers the sexual cycle. And they make a structure called the oxospore, which is actually a very large cell. And they end up regenerating a large cell with large theca. And then they go through the whole process again. Now, probably most diatoms don't have to do this. And we've got some images here. These are pictures that we've taken in the lab of a diatom that we work on extensively. And you can see that in this particular example, the center of the cell where the valves will be made is actually wider than the 2 mother cell valves. Here's another example here. This is actually an image of a diatom making new valves. You can see the 2 daughter cell valves here. They're fluorescently labeled. And it's very clear that they're actually larger than the 2 mother cell valves. So there's flexibility in most diatoms, and they don't have to undergo the size reduction process. A characteristic feature of pinnate diatoms is that many of them can actually move on a solid surface. And these are micrographs from my collaborator Rick Weatherby at the University of Melbourne. This is a light micrograph of 2 pinnate diatom species which are moving on an agar plate. And you can see that there are trails that these cells leave behind. This is an electron micrograph of one of these trails. And what Rick has shown and other people have shown is that these diatoms actually secrete an adhesive mucilage that will stick to the surface. And in fact, where they secrete it is through the raphe. And so there's an internal motor mechanism in these cells. The cells basically will stick down this sticky substance, and then there's an internal motor which pushes or pulls against that material and allows the cells to move forward. It's really a fascinating way in which these cells can move. So that's something about the basic cell biology of diatoms. And one thing that's important to consider is that diatoms are extremely successful organisms in an ecological sense. They're found in marine environments, freshwater, in Arctic and Antarctic ice, and in soils. They're incredibly successful organisms worldwide. And when you consider the fact that they're photosynthetic, it's not such a surprise of the fact that they're responsible for what's estimated to be between 1/5 and 1/4 of the total global carbon fixation. This is actually slightly more than the world's rainforest. And what you're seeing here is a satellite image of the Southern Ocean and a measurement of chlorophyll concentrations in the Southern Ocean. And diatoms are particularly abundant in this region of the world. And if you consider the vast size of this area, it's not a surprise that diatoms are really one of the largest primary producers in the oceans. They fix carbon from the atmosphere and provide a food source for many other aspects of ocean life. And of course, this process works by diatoms and other phytoplankton taking carbon dioxide from the environment, fixing it into energy stores for their own growth and also for the growth of other organisms, and then respiring oxygen back to the atmosphere. And there's a lot of interest right now in research on diatoms to understand their contribution to the global carbon budget because they're such major contributors. And it has to do with the fact that we have rising CO2 levels in the atmosphere. People are beginning to model phytoplankton contributions to carbon fixation because it's obviously an important source of fixation globally. An interesting feature about this, if you look at this closely, is the fact that diatoms and other phytoplankton are not equally abundant throughout the ocean. There are regions where they don't grow so well. And the reason they don't grow so well is the fact that they're limited by one or more particular nutrients. And there are 4 major limiting nutrients for diatom growth: nitrogen, phosphorus, iron, and silicon. It ends up that these are similar nutrients that are required for the growth of our plants, terrestrial plants as well. And it's an interesting thing to think about diatoms affecting the climate on such a large scale. But really what we need to understand more about is the fact of what's happening at the level of an individual cell. So these nutrients are actually either preventing or promoting the growth of diatoms, and they have a tremendous effect overall on the global scale. Our own lab has been interested in understanding the metabolism of both nitrogen and silicon in diatoms, and I'll be talking a bit at length about the work that we've done in this regard. This is a transition slide. I wanted to bring everyone down to the unicellular level after looking at the globe for a while. So these are just some images of diatoms. And one of the things that we're most interested in studying in our lab is nutrient uptake by specific transport proteins in cells. So in addition to the rigid cell wall that we've seen in these images before, each cell is surrounded by a relatively impermeable lipid bilayer membrane. And when a cell needs to utilize a nutrient, it has to transport it across the membrane. And the way that cells do this is they make specific proteins called transporters that specifically recognize a particular nutrient and can actually distinguish between different nutrients. And they actually form a channel through the lipid membrane and allow the nutrients to come into the cell. And this is a really fascinating process and a very interesting process as well. But there's an added bonus to studying transporters, because transporters are really a gateway for the cell's interaction with the environment. So it's a commitment step by the cell to be dealing with a particular nutrient. And as such, transporters tend to be very highly regulated. So if you take a nutrient up into a cell, you can affect a lot of processes inside the cell. For example, enzyme activity, you can activate pathways, you can actually induce signaling within the cell, and these are all interrelated processes. If you induce signaling, you can actually induce changes in gene expression. And so if you change genes, the genes that are being expressed, you can actually change the suite of proteins that are being made in the cell. And there's a feedback mechanism. You can actually go back and alter many aspects of cellular metabolism at this level. In fact, you can feedback all the way to the transporter level. And so the reason we're interested in transporters is not only the uptake process, but studying transporters allows you to get into this much more complex regulation. So if you're interested in studying how a diatom is interacting with nitrogen, for example, it's an ideal way to do that, because you're going to tap into all of this regulatory machinery by studying the transporters. And our group was the first to clone genes encoding both the nitrate and ammonium transporters for diatoms, and we've done quite a bit of work on this regard. There's an interesting level of regulation that goes on here. The ocean predominantly has nitrate as a nitrogen source, but it's energetically costly for a phytoplankton to use nitrate because it has to use a lot of electrons to get it to a stage where it can incorporate it into amino acids. There's a very general preference for ammonium by phytoplankton because it can be incorporated directly into an amino acid. And so there's a very nice regulation going on. In the presence of nitrate and the absence of ammonium, the cells will deal with the nitrate. But if you add ammonium to a culture that you're growing, the nitrate transporter is actually shut down. The gene is shut down, and we've shown this. So it's one level of regulation. We've also shown that the ammonium transporter gene is regulated as well. Not the same way, slightly differently, in actually a more subtle way. But what this is allowing us to do is to begin to tap into the overall regulation of the utilization of these nutrients by the cell. I'm going to talk now about silica metabolism in diatoms, and there's several aspects to this. Our group also was the first to clone the genes responsible for transporting silicon into the cell, the silicic acid transporters or silicon transporters. And once silicon is taken up by the cell, we know that it has to be transported through the cell by a mechanism that we really understand very little about. And it has to make its way to the special compartment in the cell where silica polymerization occurs. That's the silica deposition vesicle, or SDV. We also know from work in our own group and others that there's a feedback mechanism. In fact, the amount of silicon that's taken up by the cell is very much regulated by how much the cell needs to use to make the cell wall. I'm going to talk now about the silicon transporters more extensively, and our interest in these proteins has to do with the fact that these were the first proteins shown to specifically interact with silicon. Now, silicon is an essential element not only for diatoms but also in our terrestrial plants and also in vertebrates. It's an essential element for the formation of bone and cartilage. And yet no one understands anything about the biochemistry of silicon because no one has a protein to look at, and this really is our first opportunity to do that. And so the way this protein works is that it sits in the lipid bilayer membrane. Silicon is present extracellularly, and there's likely to be a specific binding site for silicon on the extracellular face of the protein, to which silicon binds very tightly. And it's literally like a lock and key mechanism. There's a specific arrangement of amino acids in this region of the protein that binds silicon specifically and can discriminate between silicon and other similar elements. Once silicon is bound, the protein can enable the transport of silicon across the cell membrane, and this is likely to be driven by an ion gradient, and then release it to the inside of the cell. Our interest in this protein is the fact that it's very likely that the protein is interacting with silicic acid through a cooperative interaction between amino acid side chains on the protein, which are drawn here, versus the bulk of the protein, which interacts specifically with silicic acid. And it's the specificity of interaction that we're interested in. Now, we don't know that these transporters are interacting through hydroxyl groups, as I've drawn here. And this is actually one of the goals of our research, is to understand what specific amino acids are involved. But hypothetically, this is a possibility. And once silicic acid is bound by these proteins, based on precedents and other transporters, in some cases, there actually can be a chemical reaction catalyzed, where water is stripped away from the ion that's being transported. And this is literally a chemical reaction. And we're interested in this because biology can carry out chemical reactions under ambient conditions. In fact, most of the biomineralization by diatoms occurs in the southern oceans, where the temperature is 4 degrees centigrade. If you look at industrial processing of silicon, it's done under extremes of temperature, pressure, or pH. You can't do anything with silicon in our classical chemical techniques at 4 degrees. And the reason that biology can do these things is the fact that there's a very specific interaction between amino acids and proteins and their substrates. In fact, there's a very specific coordinated interaction in 3 dimensions that lowers the activation energy for a reaction to occur. So our interest in understanding the nature of this interaction in the detail level has to do with the fact that this actually could improve the industrial processing of silicon. If we can understand more about how an enzyme or a transporter is interacting with silicon, this knowledge could be used or adapted to industrial processing. And silicon is an extremely important element in many industrial settings, not only computer chips, but coatings, paint, sealants, and polymers as well. The way in which we're going about doing this is comparing amino acid sequences of these transporters from different diatom species. So what I'm showing here is an amino acid alignment of a short stretch of the protein. And what's important are the amino acids that are colored in red. These are conserved in these 7 different species that I'm showing here. And through the course of evolution, amino acids that are essential to the function of a protein tend to be conserved. And so these are good targets for candidates that might be involved in the actual recognition and binding event. We're planning on testing this directly by going in and specifically changing these amino acids and developing a system whereby we can test this directly. And hopefully this will get us to the result of understanding how these proteins directly interact with silicon. The other aspect of silica metabolism that we're interested in is the actual polymerization event and the structure formation event. And this is a dynamic process. The SDV is actually stretched and molded during the formation of these structures. And it's quite a fascinating process. We understand a little bit about this, and I wanted to show one model. This is a model of a pinnate diatom species and the formation of the RAFE structure in this. It's known that initial deposition of silica occurs in the center of the cell in a location called the central nodule. And then simultaneously from both sides of the central nodule, there grows outward a thin silicified tube, which eventually spans the entire length of the cell. Now this tube is always encased within the SDV. The SDV is literally being stretched the entire length of the cell. Once it reaches the end of the cell, there's a curvature event that occurs, and then simultaneously from both ends and also from the middle, new tubes are produced, and they grow in towards each other and can actually meet in the middle and fuse precisely. This is really quite remarkable when you consider the distances involved in the cell. There's a very precise alignment of these structures, and you can actually see the region where they fuse together. How does this work? Well, it's known that during the expansion process, the SDV is lined by microtubules. In fact, this is an actual electron micrograph of the central nodule right here. And then sitting directly above it is an organelle called the microtubule center, or MTC. It's an organization of microtubules, and microtubules are proteins that form an intracellular scaffold in most eukaryotic cells. And these proteins form rod-like structures. They can assemble and disassemble specifically at one end, so they can grow in length and retract in length specifically at one end. They also can form tracks upon which other proteins can move. There are motor proteins that can interact with these microtubules and move specifically in either one direction or the other. And so we have a couple of models for how this expansion might occur. One is that the microtubules are attached to the end of the SDV, and as the microtubules expand, the SDV is stretched. The other possibility is that the microtubules are pre-existing tracks upon which motor proteins are moving and can expand the SDV, as we see here. We don't know which of these mechanisms are correct, but they're both things that we can test. So in addition to this large-scale stretching and molding, if you look at the detailed structure of silica in a diatom valve, it seems to be far more complicated to be explained simply by interaction with the cytoskeleton. There's too much going on. There's too much detail here. You wouldn't expect the cytoskeleton to be able to control all of this. And so we think it's very likely that major determinants of structure in diatom silica reside in the SDV membrane. So in addition to the microtubule-associated proteins, there are likely to be a whole suite of other proteins that are involved in forming these structures, and they're likely to reside either within the silica or within the SDV membrane itself. And we're actively engaged in a project to study these proteins. So we're isolating these proteins from the SDV. We can apply a variety of extraction techniques. In particular, when we extract with a detergent known as SDS, we can extract membrane-associated proteins. And this is a polyacrylamide gel separation of individual proteins from different extracts. So each of these bands that you see here correspond to a unique protein that may be derived from the SDV. And we're currently engaged in determining the sequence of these proteins. And this will allow us to identify what they are. In addition, we can also identify genes that are being expressed during the period of cell wall synthesis that make these proteins. And by cloning and sequencing these genes and then comparing the sequences, we can identify those genes that are really responsible for the formation of these structures. And this is where a major aspect of diatom research is going right now. People are very interested in this, and we're very actively involved in this ourselves. And one thing that helps us tremendously in this regard is we now have the first genome sequence of a diatom. This is not yet published. It should be coming out later this summer. The diatom that's been sequenced is Thalassiocyrus pseudonata. This is an EM of that. The genome is about 34 million base pairs in size, which actually is quite small. To put it in perspective, the yeast genome is about 1/3 of that. The human genome is about 100 times larger than that. We have a very precise map of all the chromosomes in this species. We actually know exactly where each gene is because we have an optical map of this genome. And one very interesting feature of the genome that's come out is that the gene sequences vary slightly on the order of 1%. So in a cell, you have duplicate copies of chromosomes. And so normally, 2 copies of genes are present that are very similar. Well, in a diatom, they differ slightly by about 1%. And this might explain why diatoms are so successful in an ecological sense, because they don't just have one copy of a gene. They have slight variants of copies in the gene that may allow them to adapt to changing environments better. Now, this is somewhat of a bland diatom. It's not the most gorgeous one compared to the pictures I've been showing before, but it's important to realize that we're at the very beginning stages of understanding how diatoms actually make these structures. And in fact, we've been doing some electron microscopy work on this species, and it's actually quite a bit more complicated than it looks on the surface. Ultimately, though, we'd like to start looking at species that make more complicated structures. And the idea here is to survey genes that are present in these other diatoms. And it's likely that there's a core set of genes that are involved in doing the basic reactions of silicification in all these species. And we should be able to identify those. But there are also likely to be specific genes that differ in these different species, either radically different or subtly different. And it's these differences that are going to be responsible for the formation of the different structures. And so by doing this sort of a survey, we can understand much better the role of these specific genes in forming specific structures. Now I'm going to talk about nanotechnology and the potential role of diatoms in this area. Just as definitions, nanotechnology basically is dealing with making materials or devices which have features on the nanometer size range. Nanometer is extremely small. It's about 1/80,000th the diameter of a human hair. And it's estimated that the total impact of nanotechnology will be tremendous. And the reason is not so surprising. We're not really proposing to develop entirely new technology. We're taking technology that already exists and downsizing it. The advantage of this is that less material is needed to make a particular device. Less energy is needed to run the device. We have faster processing. So we all know about the downsizing in computer chips over the years. An electron travels only so fast. If you shrink the distance it has to travel, the processing gets faster. And in addition, nanoscale materials do have novel properties. In fact, you start to see quantum level effects on the nanoscale. And this is already being used to advantage. And so typically when you see an advertisement for nanotechnology, you see something like this where we have a metallic robot sort of a thing. It's like, I think it's floating in a human body. I'm not quite sure what the grid is. It must be like a mathematician's mind or something. But you can see it's attacking this cell, which I'm certain is a cancer cell, and zapping it with millions of volts of electricity. Well, this is, I won't say far from the truth, but the development of devices such as this will take a long time. And another way of looking at nanotechnology is the fact that it's already been developed. Nature has already developed these devices on this size range. And the example shown here is the flagella in a bacteria, which allows the bacteria to move. It's an exquisite molecular motor made out of proteins, carbohydrates, and lipids. It's extremely efficient and works incredibly well. And so it's important to consider the contribution of nature to nanotechnology, because nature has had such a long time to devise these devices. And in fact, if you look at a chart of a biochemical pathway in a cell, you can't even read this because it's so complex. But for example, in the formation of triacylglycerol, there's a tremendous number of steps involved, each catalyzed by a specific enzyme that makes very little byproduct. Each enzyme is very tightly regulated. There are feedback mechanisms throughout the whole thing. Nature has had a lot of time to refine these systems. And so the other way of looking at nanotechnology is that let's use systems that have been developed already and try and adapt them for purposes that would be useful to us. And there are many possible applications of diatoms in nanotechnology simply because they make such a wide variety of materials. I'm going to talk about specifically information storage and transmission, which is basically computation and communication. This technology is already moving towards optically based processing because light can carry more information than electrons. It's also been downsizing over the years. And so what's being developed now are new technologies that blend these two possibilities together. And to give you an example, current optical technology would be a fiber optic cable. Single fiber in these cables is on the order of the diameter of a human hair. This is some new technology. You see a red light here, which is being transmitted by a silica nanowire. The wire is about 50 nanometers in diameter. And just for reference, this here is a human hair. And so the idea is, again, downsizing allows you to carry more information in an equivalent amount of space. A very promising class of materials in the development of optically based nanotechnology are known as photonic crystals. And basically they consist of materials often made of silicon or silica, which have regularly spaced pores in them, or different regions of different refractive index. And the spacing of these pores is on the order of wavelengths of light. In this particular example, there's a defect in this structure through here, and this can actually act as a light waveguide. You can actually propagate light through this material and bend it at a right angle with very little loss in intensity. In fact, you can't do that with a fiber optic cable. If you bend it too much, you lose a tremendous amount of the light that goes through it. This technology has already been used to develop micro and nanoscale lasers. And a particular intriguing development is the development of materials called photonic bandgap materials. So here again we have our material with regularly spaced pores. There's a defect here in the center. And the way in which this works is that light is shown from the back of this material towards the front. And not only does the light propagate through the defect, but there's actually a selection for a particular wavelength of light. So this allows you to control the propagation of light specifically. And so this technology is very promising as a means of eventually replacing the electronic circuit with an optical circuit when you can control the propagation of light directionally and also select a particular wavelength. The basic concepts of this technology have been developed. The biggest problem is, however, current manufacturing techniques to make these materials are two-dimensional. So it's based on computer chip manufacturing technologies where we deposit layer by layer or we etch away layer by layer. You can make three-dimensional materials, but it's very difficult to do. And is very difficult, especially to do in large numbers. And so it's been acknowledged in several reviews that the greatest obstacle to the development of this technology is our ability to make materials with the proper features, inexpensively and reproducibly and in large numbers. This is where diatoms might come in. Even though the diatom valves are on the order of microns across, most of the pore structures that we see are on the order of hundreds, tens to hundreds of nanometers, which are exactly the size we're interested in looking at for these types of materials. Diatoms reproduce these structures with exactness from generation to generation because the process is genetically encoded. We can make tremendous amounts of this material. Diatoms grow very readily. We can get billions of cells with these exact features in a week for a very inexpensive amount of money. And so what we're interested in doing is trying to test and see whether diatoms could actually serve as photonic bandgap materials. And here's another example of a man-made material. In this case, it's propagating red light. And this is a diatom. If you look at the regular spacing of pores on here, this is at least equivalent, if not better, than the man-made material. And so we're very interested in testing whether these materials can actually work in such a way. There have been theoretical calculations based on this pore spacing which support the idea that it will work. Another important aspect of the use of diatoms in nanotechnology has to do with technology that was developed by our collaborator Ken Sandage, who's at Georgia Tech. And what Ken has done is develop a technique where he can take a diatom, and these are from the genus Alkalocera, and he can convert it from its silica-based chemistry to another material. And the numbers here are just denoting nanoscale features on this particular diatom. And what he does is he takes the diatom frustrable and puts it in a high temperature chamber with, in this case, magnesium gas. And the magnesium actually displaces the silica and becomes solidified. And what you're seeing here is a magnesia replica of the silica in a diatom with maintenance of all of the nanoscale features. It's a really remarkable technique. What this does for us is it allows us to think about using diatoms as not only as a silica-based source of materials, but another source of materials. So we're combining the ability of diatoms to make these incredible structures with potentially optimizing their applications in nanotechnological, in a nanotechnological sense. Ultimately, where we're headed with this is to be able to genetically modify the structure. And the idea is fairly simple. If we have a gene that is involved in making the pore in a diatom and forms a pore which packs in a particular way, if we can modify that gene such that it makes a different shaped pore, perhaps we can actually modify the structure of the diatom. The concept here is that if we really learn enough about the process, we can actually genetically tailor these structures for a particular nanotechnological application. In addition to that, these genetic-based approaches are actually very valuable ways to understanding how particular genes are working in the diatom. If we have a gene that we suspect is involved in forming a pore but don't know that, if we modify that gene and see that the pore structure is varied, then it's clear that that particular gene is involved in the process. And so we're actually gaining in two senses where we can potentially modify structure specifically, but also learn a lot more about the genes that are involved in this process. So pretty much anyone who's worked on diatoms or seen them is attracted to them. I find this fairly universal. And the attraction is sort of twofold. One is the intellectual fascination. When we look at the sorts of structures that diatoms can make, you're initially drawn to the ideas, how can they do this on such a small scale with such precision and so reproducibly? But diatoms are really aesthetically beautiful as well. The incredible variety of shapes and structures that they make are truly amazing. The fact that they're on the size range where they interact with light is also quite intriguing. And back in the Victorian era, diatomists had gotten the idea of looking at all the different shapes and structures that diatoms can form, that they could make art out of diatoms. And there's a current practitioner of this. His name is Klaus Kemp. He's in England. And basically, he's adapting techniques that were developed a century ago where people will actually move individual diatom cells around in a microscope slide to make pictures. And he's a true master at this. These are all individual diatoms arranged under the microscope by Klaus. And I remember the day that I discovered Klaus's website. It took the whole afternoon just to look at all these things. It's truly amazing. What really is amazing is the detail. There's a headlight on this bicycle and a tail light. and then there's actually a chain here. He's also done some very beautiful abstract arrangements as well. The rosette structure are very popular. This particular one, he was proud of himself. He said, there are 251 individual diatoms in this. What a wicked feat of micro-manipulation it is. And this is actually an older slide that Klaus has gotten, something that was done in the Victorian era. It's really quite beautiful. So after I had discovered Klaus's website, I got inspired, and I was wondering whether he could make a reproduction of the Scripps Institution of Oceanography logo. So I emailed him, and I sent him a copy of this image, which has the waves and the ship and then the trident on it. And he called me a couple hours later and was very enthusiastic about it. And about 2 months later, he sent me a slide. And it's truly amazing. I have the slide, actually. When I got the slide, another 2 weeks was spent taking photographs of it. There are 3 major diatom species in this arrangement. There's the ones that make up the waves. There's the circular ones making the circle. And it's overexposed in this image in the ship, but you can see it better here. It's incredible. If you look at this, this actually isn't a diatom, by the way. We think this is a sponge spicule, but it's okay. But if you look at the detail, it's just simply amazing. And in fact, one of the things that strikes me most about this, in order to make this a nice smooth curve, he actually broke the diatoms on this part of the structure to different lengths. He's a true master at this. And I really enjoy this a lot because it demonstrates sort of where we're headed in diatom research, in the sense that the colors you're seeing here are actually true colors. So if you take a photograph in dark field microscopy, not only are these diatoms wave-shaped, but they have features that interact with light in such a way that they refract blue light. So they literally are blue. And if you take pictures of this with light at different incident angles, you can get a whole series of refractions off of it. And to me, it really demonstrates the potential of using diatoms as optically based materials. So I'll close with one more of Klaus's masterpieces. Again, hair is parted on one side here, and on the other side here, there are glasses here, and thank you for your attention. The question is, why are there so many shapes? I think, why are there so many fingerprints on a human? There's a space within which these structures need to be shaped in order to function properly, but it's actually a vast space. So nature tends to explore variety. And so within the scheme of what's needed in the cell wall of a diatom, there's obviously a tremendous amount of variety. One way to look at this is that cell surface phenomena in biology tends to change a lot. It's a defense mechanism where if the cell surface is changing a lot, it allows the cells to escape, for example, predation by something that recognizes the surface. Well, forming the wall is a cell surface phenomena, and so that might be part of why there's such a tremendous variation in the structures. Should we be thinking of these as plants or animals? This is a great question. So we now know from the genome sequence that they're half plant and half animal. So half of the genes are similar to higher plant genes and half are similar to animals. They're really quite unique. And this might be another reason as to why they're so adaptable in an environmental sense. They've got the best of both worlds. Yes, so they're one of the primary producers in the food chain. So they're basically fixing CO2 into a form where other organisms can feed off of them, yes. I don't know that that's really been accurately measured. So you would predict with increasing CO2 levels, perhaps they would be increasing, but the problem really comes with the other limiting nutrients, which in many cases aren't changing. You're probably familiar with ocean fertilization experiments with iron, for example, which induces blooms of diatoms. So it's not so much the amount of carbon dioxide that's available, but the other nutrients that are really limiting. They're in the surface waters, yes.
In the surface?
Yes, because they're photosynthetic, you need to have light for them to grow.
So you don't have to go way down for those?
No. Do diatoms use iron? Yes, they do. Most cells require iron for growth. In trace amounts, but it's very significant. They do need it for many of the reactions of photosynthesis, for example. Yes, well, they're active in the Southern Ocean because there's a lot of nutrients available. The circulation in the Southern Ocean, there's a lot of upwelling events. So nutrients are available to them. It's not so much the temperature as the availability of nutrients. So it just happens that the Southern Ocean is an area where it's rich in nutrients, and so diatoms thrive there. And they've adapted to being able to grow at those temperatures. That's a great question. There's actually a species of diatom that lives on the large kelp. And in fact, it's so adapted to living on the kelp and using carbon sources from that kelp, it's lost all of its ability to photosynthesize. It's actually clear. We have it in our culture. So as I mentioned, pinnates evolved later on, and they basically have adapted to living on surfaces. Oftentimes on surfaces, there's a concentration of nutrients, and it's easier just to move around on a surface to find them. So deposits of diatoms globally are regions such as the Southern Ocean where diatoms have been abundant for many, many millions of years. So as a diatom is growing, as diatoms are growing and die, they'll actually begin to sink through the water column. Now, there's actually a recycling process that occurs, and it's estimated that silicon that's incorporated into a diatom is recycled about 40 times before it finally makes it to the ocean bottom. And don't ask me how those calculations were done, but we seem to believe them. And, but eventually they do sink out. And so when you see deposits of diatomaceous earth, it's a region where diatoms were able to grow very abundantly for a long period of time and have just settled out in those regions. The largest size of diatom, I believe, is a rhizoselenia. It's about 1 millimeter in size. That's very rare. Most of them, again, are quite small. Yes, diatomaceous earth is used in filtering. Yeah. In fact, many materials scientists know diatoms only because of that. The question was, it was more of a statement. It was that diatomaceous earth is used as a filtering material. And yes, it is. It's also used as an insecticide. So apparently it's irritating to the cuticle of insects, and so you can spread it on insects and they don't like it.
Well, I suspect, as usual, there'll be a number of you that will want to come up and chat with Mark afterward. But Mark, thank you very much. That was fascinating. Really.